So let's say you are at a crime scene and you are investigating a murder, and the only piece of evidence you have is this drop of blood which you have nicely packed into a test tube, and you want to accuse one of these two suspects. You have two suspects and you want to know which of them did it. So you have just this drop of blood, and you have to figure out a way to get to the bottom of this basically. And you might remember from bio class that blood contains DNA. So what you do is you subject your sample to a process called DNA extraction, and you can get the crime scene sample into a long strand of DNA. And say you can do something similar for each of the suspects. You draw their blood and then you extract their DNA, and you get this nice long sequence of DNA for each of them. What can you do then? Well, if you remember, then each DNA sequence, each person has a unique DNA sequence. So what you could just do is you could do a pointwise comparison, nucleotide by nucleotide, to see which one of these has the exact same sequence as the one you found in the crime scene. Any one of these two will have an identical sequence, and the one that matches exactly is our criminal. Okay, that sounds nice, but there is a problem, and the problem is that sequencing is complicated. Not only is it complicated, it is expensive, and also we have a kind of a historical problem here, which is that sequencing for human genomes was only possible post 2000s, whereas DNA fingerprinting as a technique to figure out who the criminal is, or to settle parental disputes, this was something that was discovered in the 1980s. So this video is basically about how on earth did people figure out back in the 1980s, before they had advanced sequencing methods for DNA, how on earth did they profile a person's DNA and use it to basically solve a murder mystery that you might find on TV and so on, right? And the broad strategy to do this is looks something like this. So you use restriction digestion to build a unique size profile of DNA fragments. Then you separate the fragments with electrophoresis and you visualize it. Okay, so now I don't expect you to understand this entire sentence, but what you should know for the sake of this video is what this means and what this means. So if you don't know what restriction digestion or electrophoresis mean, then you should probably watch the specific video for that. Okay, so for the sake of this video, I'm assuming you're familiar with those two techniques. So the first step is you do restriction digestion to break the sequence into a unique size profile of fragments, and what you do might look something like this. So say this is the DNA sample you got at this crime scene, and your restriction enzyme would cut it at specific sites, and if one of the two suspects have an exactly matching DNA, then treatment with the same enzyme will cut their DNA at the same site. Now bear in mind that the human DNA is pretty big, so it's going to cut in way more than just two or one site, but for our purposes, for now, let's just assume that it looks something like this. We'll get to the size issue later. Okay, and you could follow that up with gel electrophoresis. So if just for a quick refresher, what gel electrophoresis is that you can load your DNA here and you can subject it to an electric field across agarose gel, and the DNA will travel in such a way that larger fragments are held back and smaller fragments travel further. So this is a neat little that's a funny spelling mistake, but this is a neat little separation technique for DNA that allows you to separate the larger fragments from the smaller ones. So what does that look like if we subject these three samples to gel electrophoresis after restriction digestion? Well, you take a look at the crime scene sample, and what you'll have is you'll have a small fragment. So let's draw that here, and you have about two medium-sized fragments. So let's say you have one here and one here. Okay, and let's look at suspect A's sample. Well, suspect A after restriction digestion has a really long sample and a kind of smaller samples. So I'm going to draw this one here, and the larger fragment comes all the way here. And as we see, suspect B seems to have the same cleavage pattern as the crime scene sample, so that's going to look like this. Okay, so you have a agarose gel separation pattern that looks like this. Keep in mind that if you just do a gel separation, you still won't be able to see the DNA because well, DNA is still tiny. You haven't changed any optical properties of it, but typically you have these staining methods, these dyeing methods for DNA. You might use something like ethidium bromide, or some chemical that basically binds to the DNA and lets you kind of visualize it, so that you can actually see the bands and they're not just like invisible and under the gel. Okay, so it almost seems like we've solved the issue, right? It seems like we have this nice little barcode creating technique to identify individuals with the same DNA. So clearly this seems to be a match between here and here, right? So is it that are we done? Are we done? Is this how DNA fingerprinting is? Is there all to it? Well, it turns out no. And the reason is that we've been ignoring one huge complication in all of this. And the complication is that DNA, or rather human DNA is massive. Okay, so if you remember, human DNA is in the order of 10 to the 9 base pairs, so that's like billions of base pairs long. And restriction site, as in the sites at which the restriction enzyme will cleave the DNA, they're typically much shorter. They're like a few base pairs long at most, right? So what does that mean? So that means that if you have a long DNA strand of DNA, double-stranded DNA, it's not a single strand. I've been saying strand all this while, but if you have a long sequence of DNA, as long as the human genome, then and you treat it with a restriction enzyme, then it's going to just keep making cuts all over the place, right? It's going to make so many cuts, just because of pure chance, because the small restriction site is because of pure chance is going to occur so many times, and I might as well just shade it with blue. There's going to be so many cuts at this resolution, right? So and the reason that's a problem is because when we actually subject this to a gel electrophoresis, instead of seeing this clean nice signal, what we're going to see is a very uninformative smear. It's going to look like this, we're not going to get any nice little bands. We're just going to get this crazy smear that's just all consuming, and it's going to be the same for each of our samples. Now if that wasn't bad enough, we have a problem that's almost like, that's almost like, working in cahoots with this problem, and that is that 99.9% of human DNA is identical. I haven't written the word DNA, so let me just write it. DNA. 99.9% of our DNA is identical. Now that's a problem, right? Because that means that not only are you going to get these smears, they're also going to be 100%, well, not 100%, but they're going to be mostly identical. They're going to be, any differences is not going to be visible to the human eye. And so it seems like our approach to this entire thing seems very naive right now, right? So how do you actually get around this problem? And the secret to this is basically to kind of zoom in to this 0.1% of the DNA that actually differs among people. Okay, and this is why we cannot stop at step 2. We can't just do dead gel electrophoresis and use a generic DNA dyeing agent. What we need to do is we need to be a bit clever in the way how we visualize our DNA. We need to basically specifically find a way to specifically zoom into this place, and so that we only visualize some part of this 0.1%. And that's exactly what we do in step 3. And step 3 is a technique, I should write the name. So the technique is called Southern blotting. Southern blotting. Okay, so Southern blotting lets us selectively visualize parts of the DNA that is constant in sequence but variable in size. Well, strictly speaking, sudden blotting lets us visualize any sequence of the DNA that we know, but in this particular usage, what we do is we use it to target a sequence that are constant in sequence but variable in size. Now, I want you to take a look at this combination of words, like this half sentence, and think about whether it actually makes sense, because it actually doesn't. Okay, so what I really mean by constant in sequence and variable in size, it should bother you that this is written this way, because if I give you a sequence that is CACCTG, so this is a constant sequence, well, how could it vary in size? Because it's the same sequence, right? It's 6 nucleotides long. You could even go as far as to tell what the molecular weight of this sequence would be like. So how could it vary in size? And the answer to that is, that's where something called variable number tandem repeats come in. Okay, so what VNTRs are is they are a known sequence of DNA that repeats over and over. The number of times the sequence repeats itself varies across people. What does that mean? So let's say there's a person A and there's a person B, and there is a sequence CTGA. And it turns out that there is a site in all our DNA where a sequence that's similar to CTGA or something else just keeps repeating over and over and over. But the point is that the number of times the sequence repeats is different across people. Now that's important, right? Because if you think about it, what sudden blot allows us to do, it allows us to target a specific sequence, and it allows us to basically, like, long story short, it allows us to basically bind something to it, bind something to this sequence that will let us view it under that lets us view it under the appropriate lighting conditions and so on. Yeah, and the idea is that you can target this sequence in both these people, and what you do is you cause this to is you specifically visualize the sequence, and if you notice, these two sequences are going to show up in different bands in our electrophoresis, because this is smaller and this is larger. What that means is person A is going to have a lighting pattern that after you do sudden blotting on this gel, person A is going to have a band that appears here, whereas person B is going to have the same band a bit higher, right? And the same thing is for our crime scene sample, which means that things aren't really looking that good for person B. And really, this is like the final ingredient in getting a pattern like this. Typically, you don't have just one of these VNTRs, you use many of them, which is why you'll get like more bands and so on. But the general strategy is this. Thank you for watching.